know that carbon dioxide has never been above 300 parts per million for hundreds of thousands of years, and now it's well over 400 parts per million, and that is one of the main drivers of climate change. We also know that these carbon emissions are not distributed equally. So the 10% highest emitters are people mainly living in high-income countries like the US, Western Europe, including the UK, of course, and increasingly some of the emerging economies too, China as well, has increased its emissions over recent decades. And these high emitters are responsible for about half of the 50% of the world's carbon emissions. And you can see that the poorest populations, the bottom 50%, are only responsible for 12%. So there's a big issue here of climate justice or climate injustice, because those populations that are suffering worse from climate change are the least responsible for the emissions. But we also know that although carbon dioxide is very important and it remains up in the atmosphere for, on average, over 100 years, once you put it up there, there are also other climate active pollutants. One of them is called black carbon. It's that very fine black particles you see from old diesel engines, or when people burn solid fuel in cook stoves, for example. That produces black carbon, which is also a climate active pollutant, and we can breathe it in. It damages our health, but also contributes to global warming. Methane, which is produced by natural gas, by livestock, by cattle, for example, and from landfills. And that's a very powerful greenhouse gas which remains up in the atmosphere for about 12 years. And we know that through a combination of complex interactions, this substance, tropospheric ozone, is produced, and that's a greenhouse gas, but it's also damaging to our health. And finally, man-made chemicals called hydrofluorocarbons that are in air conditioning and refrigeration also make a contribution. So it isn't just carbon dioxide, but carbon dioxide is the most important gas. So as the temperature of the planet increases, you can see the map shows you that it doesn't increase uniformly. Some parts of the planet warm up more quickly than others. So the Arctic, for example, is warming more quickly than other parts of the planet. You can see also there's a red area over West Africa, which seems to be warming more quickly as well. And on average, the land warms faster than the ocean. So the oceans help to cool the planet. You can also see from this slide that as the temperatures increase, then the risks increase. So ecosystems are threatened. They can't keep pace with increasing temperature. You get extreme storms, of course, floods, droughts, wildfires. You may get collapse of the Antarctic ice sheets, for example, which would further drive up sea level. Coral reefs die off. We see coastal flooding, river flooding, effects on crop yields, and of course a range of effects on human health as well, both direct and indirect. And these are summarized in this infographic, which tries to summarize rather a complex picture, but what it shows you is how increasing levels of carbon dioxide and these short-lived climate pollutants that I've mentioned, through a range of different demographic, socioeconomic, and other environmental factors, these influence the size, the magnitude, and the pattern of risks from climate change. You can also see that there are a range of exposure pathways, right from the most obviously direct, like extreme heat stress, for example, or other extreme weather events, through to the effects of air quality, for example, of wildfires, water quality and quantity, both floods and droughts, food supply and safety, the distribution of vector-borne disease, like obviously malaria and dengue, for example, and the effects on social factors. So as you heat the atmosphere, it becomes more difficult to work, of course, and that will drive more people into poverty because of the extreme heat exposure. And some of that work is going on, of course, in the Gambia, some of that research. So one of the questions is, to what extent is climate change affecting health now? And this is a recent piece of work led by my colleagues at the London School, Antonio Gasparini in particular. And they've been collecting data from over 700 sites in over 40 countries. And they showed that more than 30% of the total heat deaths over recent decades can be attributed to human-induced climate change. So they wouldn't have occurred in the absence of climate change. They're not due to just natural fluctuations in temperature. But you can also see from the map that there are big gaps. The grey areas are where we don't have good data, including in West Africa. So there are big gaps in the data, and the evidence that we have so far is probably an understatement of the true magnitude, true size of climate change effects. So that's just one example, how climate change increases the risk of heat-related deaths amongst adults in particular, the elderly in particular, but other groups as well. And this slide shows you how the spread of increased heat stress occurs as global average temperature increases. So once you get a temperature increase of, say, over 2.5 degrees on average, then about 1 billion people will be exposed to such extreme heat that it prevents even moderate physical activity safely in the hottest month of the year. So that shows you how increasing heat stress is damaging the prospects for health in many parts of the world, including, of course, in West Africa. And the work that's going on at the moment, led by Anna Bonnell and others, and supervised by Andrew Prentice and myself, is documenting the effects of extreme heat stress on pregnant women subsistence farmers. And we're finding that they are exposed already to very high levels of extreme heat, which is having effects on the fetus. We also know that wildfires have an effect on human health. This is a recent paper in Lancet Planetary Health, and on the right you can see how all-cause mortality, cardiovascular mortality, respiratory mortality increases in a few days after exposure to wildfires. So wildfires may have long-term effects on human health, but they also have short-term effects by increasing death rates from these common conditions. We believe that wildfire particles are smaller than those in the particulate matter from common urban sources, and they may be more damaging. They seem to contain more oxidative and pro-inflammatory components than kind of standard urban air pollution, if you like. And some of these levels of pollution can be extraordinarily high, as shown on the map at the bottom left of this slide, some of them going up to over 200 micrograms per cubic meter, which is a very, very high level of pollution indeed. Of course, climate change can affect distribution of a range of infectious diseases. I think we may hear a bit more about that from Chris Murray and other colleagues later. But climate change affects distribution of vector-borne diseases, including malaria and dengue, of course, food-borne diseases, a range of waterborne diseases as well. It does so by influencing the pathogen, the disease vector, and of course the host susceptibility to infectious diseases, for example, as a result of impaired nutrition or forced migration. So there's a range of complex pathways by which climate change increases the risk of a range of different infectious diseases. And this is just one example from colleagues at the London School, a paper that they published last year in Lancet Planetary Health showing that there could be 4.7 billion additional people at risk by 2100 due to rising temperatures and population growth if we have a temperature increase of over 3 degrees compared with perhaps 2.4 billion if there was a temperature increase of about 1 degree, which of course, as I've said, we're already at. So it's unlikely we can keep it to that level. So it does show you the potential numbers of people that could be affected by these common vector-borne diseases. Climate change can also affect food security, and the map here shows you those countries that are likely to experience a decline in crop yield, an increase in food insecurity over coming years, the red areas, and the green areas are where crop yields may increase, but we don't know for how long those increases might occur. We also know from the Ukraine war that there can be other factors that can affect the availability of crops for export from temperate regions. So what it means is that food insecurity will increase. And on the left side, you can see the average of many of the climate modelling studies, which show you how crop yields will decrease increasingly from the 2030s onwards. And these decreases probably will be more than 10%.
Yes. Yeah, there we are.
Prof, carry on, please.
Okay,
Hello,
fine.
Prof. Are you hearing us now?
Can you hear me now?
Hello, Prof.
Can you hear me now?
Yes, Prof, we can hear you now.
Great. Okay, well look, my previous slide just showed the effects of climate change on crops, and I won't say any more about that. Just to emphasize though that climate change is likely to have effects, or is having effects, not just on physical health, but also on mental health as well. And we're increasingly understanding that when people are exposed to extreme events like floods and droughts or wildfires, extreme storms, they suffer not only the physical effects, but also the mental health effects, which can persist for long periods. And there are new concepts like this idea of solastalgia. Solastalgia is the kind of distress that is caused by environmental change, when communities see their familiar environments being destroyed by climate change. So, for example, melting ice in the Arctic or extreme droughts in Australia have been documented as causing extreme distress in a range of populations. And we also see that young people, particularly, are suffering from climate anxiety. So they're anxious, they're worried about a potentially hazardous and uncertain future, and that's totally understandable. We need to quantify and understand the mental health effects of climate change as far as we can, and there have been a couple of reports which are shown on this slide which summarize some of the recent evidence. We also know that
Yeah, it's back now.
Prof, can you hear us now?
yes, can you hear me very clearly? Can you hear me?
Go ahead, please.
Yeah, so the map shows you where there are a number of hotspots experiencing a combination of several extreme threats due to climate change in much of Africa. You can see, for example, parts of West Africa like to be suffering from sea level rise, other parts of Africa suffering from desertification, decreasing rainfall, particularly in that belt going eastwards from Dakar. You can see how the Nile Delta, for example, is experiencing, like to suffer from sea level rise, as well as decreasing rainfall, the effects on the flow of the River Nile, and many other locations within sub-Saharan Africa where there are like to be multiple climate impacts. And these will have synergistic effects on human health. This will lead most likely to migration, both internal and external. A World Bank report a few years ago suggested that there could be, by 2050, perhaps 86 million more climate-related migrants in sub-Saharan Africa, and that's probably a very conservative estimate of the effects of climate change on population displacement as a result of declining crop yield and declining water availability and coastal erosion. We also know that the outcome of the COP26 conference last year in Glasgow held some advances, but many disappointments as well, and those are being discussed at this very moment in Egypt in COP27. But the rich countries did not come up with $100 billion that was agreed as early as 2009, and that goal is not likely to be reached until 2023. There was no agreement on phasing out coal or fossil fuel subsidies, and many of the richer nations refused to accept liability or provide compensation for loss and damage from climate change. And so these will be very big topics for discussion at COP27. There may be some slight changes in position.
Yes, we can hear you. Those.
Okay, so we can...
From there in is not from here. Yes, we need to connect it. Yeah, back.
No?
Please go on.
change. We need more resilience.
Hello, Prof.
Yeah.
Can you try again? Yeah, please go ahead.
When electricity goes off, it takes time for all those things to come up again. That's the problem. And this one, the speaker is not meant for a
Can
big
you
concert.
talk to him so that he understands that we are having challenge?
Hello, Prof. Can you hear us?
I can.
Yes. Yes. We're having some challenges because our room system, the power is very erratic. It keeps going off and on. But we just want to try last time. So just bear with us 30 seconds and then you are good to go. Again. We're very sorry.
No worries.
Okay, thank you.
Can we know how many slides more do you have to go so that it's possible?
Okay, I'm sure he's
almost done,
he's
you know.
winding up.
This way.
Let's just make sure.
Prof, you can start.
So, as I mentioned, we can either adapt to climate change or we can mitigate, and we both have to adapt and to mitigate emissions as well, particularly in high-income countries. And to give you one example of adaptation, this is the example of community heat action plans. This is to try to reduce the health effects of extreme heat exposure. And this requires an integrated response involving the health system, but also other sectors as well. So, for example, there may need to be an early warning system for very extreme heat exposure, community response plans, which could include distribution of drinking water, potable water, the targeting of vulnerable groups, the increasing use of green space to cool cities, for example. We know that vegetation helps to cool urban environments in particular. So it's a combination of different sectoral policies that can help to reduce the health effects of extreme heat exposure, including the effects on adult mortality and on more vulnerable groups like pregnant women as well. And I think there's a big research agenda here for the Gambia in evaluating potential adaptation strategies that can help us to withstand more effectively extreme heat and other extreme events due to climate change. But there's also big benefits from moving towards a green and clean economy, because when we burn fossil fuels like coal, for example, or oil, we create large amounts of air pollutants. So if we generate electricity using clean renewable sources like photovoltaics, then we have the benefits of the electricity without the air pollution and without the carbon dioxide that's causing climate change. And the map on the left of this slide shows you where those fossil fuel related air pollution deaths occur. You can see many of them occur in India, China, Europe, North America. including, of course, the use of renewable energy, but also more sustainable supply chains. So, in conclusion, climate change poses very many threats to human health. We can adapt to climate change as much as we can. We can also cut emissions, particularly high-income countries, and that will produce big benefits to health in the near term. So we have tons of carbon dioxide left, and that's only about, it says 9 years on this slide, it's probably less than that, it's probably less than 7 years, but all countries will have to pay their part. But how we spend that remaining carbon budget, of course, is going to be a very important question for human health and for the environment more generally. I'll stop there and I'll hand over to Andrew to his presentation. Thank you very much.
Thank you. Thank you, Prof. I think we've all had it together that we'll just pause for a while and entertain few questions. Okay, thank you, Prof. Andy. We've all had him together that climate change happens to be the biggest threat. You're fine with you.
Yes, I think you can come over here. The mics are working. You need to come over here and use this mic. You can stand in front here. Hello, Prof. That's a wonderful talk despite the challenges. Glancing through your slides, I'm not really an expert, but I'm more of an infectious disease bias. So with this heat waves and the melting of the ice and all that, is it possible that we could be having pathogens that are really not new to the current population that we have on the planet? And are there studies that are going on to survey some of these pathogens so that we can prepare and not be relaxed like the way the SARS-CoV-2 caught the whole world? Thank you.
Thank you. That's a wonderful question, and really excellent, and absolutely, you're absolutely right. There have been now a couple of workshops, a few papers, looking at the potential for emerging diseases from the melting of the Arctic ice. Because what we do know is that there are many organisms trapped within the ice, organisms like Clostridia, Toxoplasmodia, and probably smallpox as well. And we know that there are many organisms that have been formed that use
a lot in terms of stunting, the red line. We've halved it. But the bad news is that we've still got half of it left to get rid of. So we're scratching our heads why, despite all the interventions in Canabar, both health and hygiene and food interventions, we've still got so much stunting and undernutrition. And the answer was a somewhat surprising one. We looked at a natural experiment where we realized that by virtue of having so many MRC staff in Canabar, we had a huge gradient of socioeconomic status and income. And we asked for volunteers from both staff and the village, and we mapped stunting. We mapped stunting onto socioeconomic status. The red box there shows the MRC compound, and we had some staff who were living in the compound and other staff who were living outside of the compound. All those MRC staff were in socioeconomic status 1, but when we did the analysis, we saw that it disaggregated, that there was a lot of variance in 1. So I'll now show you the data after we separated, turning the microphone off instead of advancing the slides, when we separated it into these groups. So the first group on the left is our staff members who had the privilege of living inside the MRC compound. Their nutritional status was very good. If anything, it was rather better than the WHO standard. And the other groups were all very uniform. And the distressing thing is that even in group 1B, which are well-off people, the nutritional status of their children was not good until they moved into the MRC compound. So what this tells us is that there's a very high threshold to be overcome before we're going to get rid of malnutrition in Africa. And that is now well recognized, that this is not just about food. We have to try and make both what's called nutrition sensitive as well as nutrition specific interventions if we're going to mend the problem. And that is now well recognized by UNICEF, by the UN, who just last week put out this One UN for Nutrition, trying to integrate it all so that we make better efforts in all of these: social protection, education, water and sanitation, health, as well as the food system. So those are just a few of the challenges in relation to what's going on in the continent. The good news, I'm a great optimist. Africa is developing. And you see, I just took this from the TAF website as to, what is being offered. Africa is developing fast. I'm a huge optimist for Africa. We will get there. Nutritional status in Africa will improve. It'll improve relatively rapidly, I think, in the coming decades. But then, we have another concern. There are so many conundra that we do not understand that we've really got to get back to some basics in this. I've told you at the front that it's all terribly simple, but actually, of course, it's not. So this has been what has driven the work we've done for many, many years now, and I'm pleased to say that in many aspects it has yielded dividends. And I'll give you a few examples of those in the coming minutes. The system's stuck.
Controls room, can you check the monitor again? It's not changing, the slides.
Maybe because it's a big file. Okay, the other element of this is that we try to integrate our research over what's called the life course. Recent discoveries in nutrition have shown how very important this is. We call it the DOHAD, Developmental Origins of Health and Disease theme. Inherited from father and mother. From father and mother, and which carry messages as to the exposures that father and mother have received during their lifetime and even during their parents' lifetimes. Those are mostly scrubbed off. So the epigenome has to be cleaned up in order to become pluripotent so that we can create a new organism with all its different cells. But not all of those are washed off. So one implication of that is that there's the potential that we carry with us messages that have come from prior generations that may still affect our health. I'll carry on. The work we've done has been really boosted by the fact that we have this incredible experiment of nature here of seasonality. We have a dry season and a hungry season, and a wet season, which is often also called the hungry season. And the nature of the foods that people eat, and particularly pregnant mums or about to become pregnant mums eat, changes. So we reasoned that that would be, as I've just said, an experiment of nature, and we could look at the epigenome, the methylome, of babies who were conceived in different seasons. And when we did that, this was work starting about 10 years ago, something remarkable emerged. Here's one of Matt's figures from a recent paper, and the story in short is that if we know where to look, we can find that certain CPGs, certain regions of the genome, the amount of methylation on those is highly sensitive to the season of conception. To such an extent that if we plot on the right-hand side here, the maxima, the time of year, you see on the y-axis is the time of year, month of conception. If we plot the maxima, methylation maxima against that, and if we know which groups to look in, which the red ones are called metastable epialleles, I'm not even going to try with timing going as it is to explain what that is, and the blue ones are called season of conception CPGs. If we know where to look, we can find regions that are highly susceptible to season and, we believe, to nutritional changes by season. We believe, or a possible hypothesis, I think it would be a bit too strong to say we believe, a hypothesis is that these regions may have evolved to sense what's going on in terms of the nutritional environment, to record that information on the genome, and to adapt the phenotype accordingly. So this is a story that is well known, and it's possible that we have at least started to uncover the mechanism by which this occurs. Now, a recent paper by a very talented PhD of maths, Maria Derekshan, has also just come out in nucleic acid research. And the story here, you don't even need to try and digest what's in the graphic there. The story here is that if we start to dig deeper into these, they all start to fit together in terms of telling a story as to why biology has chosen these regions. And Matt's just been telling me that they have very interesting potential implications in terms of regulating metabolism, particularly in the recent example that's really coming out is in terms of the formation of the pancreas. And putting this work together with other work that's been done in Cambridge, it looks as if we may be on the forefront of looking at, on the cusp of finding some really important basic biology. Now, that's the sort of showpiece, and great credit to Matt and his team. You then ask, well, what does this matter? Do these things really do anything? And here's just one example from Toby Candler, who recently graduated with his PhD, which shows, yes, they do. Again, because our time is short, I'm not going to talk you through this story, but the top right-hand graph just shows you that there's a huge range in variation at this gene PAX8, of methylation of PAX8, and the bottom graphic tells the story that the ones in blue compared to the ones in red, in other words, whether you're hypomethylated or hypermethylated, this feeds through to an effect on the thyroid in terms of thyroid size and in terms of biological measurements of thyroid function. So the point of just putting that under your nose is to say it's almost certain that a lot of these are going to feed through to important biological and health functions, and we have many more examples. Now the next question is, well, what's the point of doing this? Can we actually intervene? And we believe that the answer to that will be that we can one day intervene. For instance, here we've done a small trial where we've endeavored to manipulate these pathways, these metabolic pathways, which are the driver of methylation. So the bottom left of that in red are how the methyl groups are supplied for the DNA. The blue circles homocysteine. Homocysteine is, if you like, a toxic metabolite which builds up if these processes are not moving fluidly and if they don't have the nutrients to drive them. So what we did in this trial was to see if we can reduce homocysteine levels with micronutrients and with a special micronutrient mixture that we designed. And the quick answer is, you can see, yes, we would be able to modify it. So you might then say, well, are you guys getting onto trials to modify it? And the answer is not yet, because we don't know what would be the optimal level of methylation. And indeed, it may be the case that if we cause children to be more methylated in the very early embryo, they may be better off in terms of some diseases, but worse off in terms of others. So although we can manipulate this, we're not yet at the stage where we'd be ready to do that human intervention. And in order to try and get ready for that, we're having to do a lot more very basic data collection and investigations to see if we can understand the pathways more. This is Matt's Eden study, which is currently ongoing in the Kenabar and West Kiang region. And the good news about this is that although this is a sort of little finding that has miraculously come out of this experiment of nature, we're pretty convinced that this is going to be important for every conceiving family in the world once we understand it better. My next little example is about cognition and development. I'm going to be very quick here. Obviously, we want to improve human capital by maximizing the very early brain development and cognition of children in Africa. That's a given. If we look at the head growth of children in Kenabar, we see that they start off life with rather small heads. So this is the percent with a low Z score. So it's, if you like, a measure of stunting, but in terms of head circumference now. So they're hypocephalic, but this gets worse during the first 2 years of life. And we would impute that this is not a good thing. That's the reference population, and we are undertaking the Indigo study, which is supplementing mothers both in pregnancy and in very early postnatal life, so 0 to 6 months, in order to try and improve brain development and cognition, which we're measuring by a number of different metrics, as you can see on the graphics there. My third example, and I'm conscious of time, so I'm going rather quickly, is in relation to iron infection and anemia. Where does the discovery science come in here? It comes in terms of this favorite molecule of ours, hepcidin, the master regulator of iron metabolism. This was only discovered 20 years ago now, but by trying to measure and understand this in the settings that we have here, we have had really important insights into why children are not amenable to iron interventions in the way that we would hope they would be. And this is just one example where we've discovered that respiratory infections appear to be a major driver of anemia, which obviously is something of a surprise. Here's the anemia rates in our West Kiang biobank. Boys in blue, girls in pink, against the African-American 5th centile. And you see in early life that they're way below that, the average hemoglobin levels, and the boxes show the percent anemic. So we have 91% of the kids are anemic. Imposed on here, superimposed on here are the expected response to iron supplementation based on the meta-analysis by the World Health Organization. So the story here is that even if we did this under randomized controlled trials, we would only get back a tiny bit of, we would only improve hemoglobin by a very small amount. So the question is why, and a lot of the answer to that is related to infections and hepcidin, which I've just mentioned. But we have just unblinded a trial which shows great hope. This is when it comes. Oh, sorry, before that, just tell you. Let me put the context to this. You'll be aware that this WHO recommends exclusive breastfeeding to 6 months. We are huge advocates of exclusive breastfeeding to 6 months. And the country, and our next chairman, Amat Bar, who's sitting over there but will be here soon, has been fantastic in terms of driving this through the National Nutrition Agency here in the Gambia. Back, please. Back one. Thank you. The problem is this, though, that although we are supporters of that, it's inescapably true that breastfed babies become very iron deficient, extraordinarily iron deficient, as you can see from a number of different cohorts here. And this is measuring serum iron, and the green panels show the reference range. So although born with a relatively good iron endowment, their serum iron plummets when being exclusively breastfed, because breast milk has extraordinarily low levels of iron in it. So we've recently done a trial with the cooperation and agreement of the National Nutrition Agency that we can intervene with iron early, which is incidentally allowed by the WHO protocol for exclusive breastfeeding. And we have supplemented babies from 6 weeks to 6 months, and this is the work led by Mamadou Ba and Carla. And these are the results from this trial, as yet unpublished. We're hoping to submit them very soon. In terms of effect sizes, and you can see that all of the measures of iron status have really improved very well indeed. The ones on the right-hand side, negative values are equal better iron status. So we've had really a very powerful effect on their iron status, much more powerful than we will have in later age groups. So we're very enthusiastic, excited about this, and we will be looking for funds to do a large-scale trial in which we would measure functional outcomes of cognition and immune development. My final little bit is this. It's a plea to integrate genetics into nutrition research. We've had a lot in this conference, and we'll have more tomorrow, about pathogen genetics. Apart from Jolyn saying that she was going to do a GWAS, there's been no other mention of human genetics. And we're trying to really ramp this up in the work that we do. And we currently have some proposals going through the Ethics Committee. It's a slow journey because the Ethics Committee are understandably, and I think rightly, concerned about how we might be sharing genetic data, but hopefully we will get there. And there's a huge imbalance here between the investment in genotyping in Europeans, so this is just an example for the UK Biobank. Almost 500,000 of people have been fully exome sequenced, and there are many papers, important papers, coming out of that as a result. If we look at the investment in African genotyping, 2% of the genomic data that goes into GWAS studies comes from Africa. Despite the fact we know perfectly well that Africans are much more interesting in genetic terms and are likely to give great benefit because of the greater haplotype diversity if we included. So arguably this should be absolutely the other way around. And here's just a little final graph. It's just an exemplar of something that we became aware of just pretty much a week ago, but I think gives a very good example of why we might want to do this work. So there is a series of genetic variants in the ApoL1 gene. And if you carry two, if you're homozygous, well, there's two variants that make you susceptible to nephrotic disease. If you carry two of these, either G1G1, G2G2, or G1G2, you're highly susceptible to nephrotic disease and ultimately hypertension, and to such an extent that you'll need a kidney transplant or to be on lifelong dialysis. The story goes that this has evolved because it's protective against trypanosomiasis gambiense. And that's why you've got this huge concentration of the gene variants in West Africa and not in the rest of Africa, a bit in South Africa as well. So here's a story where Modu, after the break, will be telling you that we have a big gap in the cascade of care between people who are on hypertensive treatment but are not responding to their treatment. So what's going on in that large number of people who are non-responders to treatment? This work, which has all been done in African Americans so far, would tell us that at least a chunk of those will be highly likely to be down to this particular variant. And if that's true, then it gives us the possibility of using these African-specific disease variants to look for African-specific therapeutics. And we're already in discussion with a company that is looking to do a trial of a therapeutic that can combat the genetic defect in ApoL1. So I'm just really putting that out there as a way of putting some substance to the plea that we need to up the amount of genotyping, human genotyping that we're doing here. So thank you. Sorry that was so disturbed and broken. It's not been a great session. No fault of yours, Mr. Chairman. Huge thanks to many colleagues in the theme and to others worldwide. And many, many congratulations to MRC on getting to the 75th. Thank you.
Thank you, Professor, for giving us those exciting research in Africa, particularly in the Gambia, nutrition research. So because of time constraint, we will allow only few, let's say one or two, feedback questions and so on and so forth. So who wants to go first? Yes, you can go first, please. Just be very brief. We are having time constraint. Yeah, go ahead. Yeah, go ahead, go ahead.
So, really great presentation.
Thank you for your question. Our questioner actually chairs the DSMB, so he knows the answer to his question. Kamija, thank you very much. I also want to thank Professor Kamija Firi for accepting our invitation to be our nutrition speaker, and you'll hear him after the break. No, this trial was completely underpowered to look for that, but thankfully we had no negative effects either on comorbidities or on growth, which, as you know, there's been suspicions that if you give iron early, you may actually slow the growth of children.
Okay. For the very nice presentation, it is really, really exciting presentation because you cover a lot regarding the stunting and also the nutrition. I have a very quick question regarding the microbiome. Did you look at the microbiome when you are doing your clinical trial, where you have the control and also the supplementation to see whether the microbiome is a little bit developed or not in the stunting children?
Forgive us, it's very hard to hear, it kind of echoes here, but I think you're asking, did we look at the microbiome? And in the iron trial, is that the question? Yeah. We haven't done that yet. Those samples are being prepared for shipping and analysis here as well. It wasn't a real aim of this particular study. It will be an important part of future studies. And as you know well, there are a lot of stories around the possibility that iron, not the possibility, the actuality that iron does cause a dysbiosis and changes the microbiome.
Thank you.
Good afternoon. Without wasting much time, I call my session presenters.
So we'll just pause a little bit and, you know, anemia, as I've mentioned, knee-jerk reaction, give iron. But if we pause a little bit and look at the iron metabolism, let's just talk about where iron is in the body. So most of it is in a ferrous functional iron, so ferrous iron, and most of it is in the red blood cells and also myoglobins. While there's another group of iron that is actually bound to transferrin receptors and is being transported in the body. There's another iron that is actually stored in reticular endothelial cells, and that is either in the bone marrow or also in the in the hepatocytes, so in the liver cells. So I will hopefully just continue. I think one big problem is that pathogens that actually cause diseases, infectious diseases, also need iron to actually grow. So the bacteria, parasites have actually developed different ways in which they try to wrestle off this iron from the host. And it could be from the transferrin that is, as it's being transported in the blood. But we believe that a lot of the parasites and pathogens actually get the iron from the intra-erythrocytic pool. So that is right within the red blood cell. There's very, very little data that actually has been done or is available in this area. So for people, young researchers, please, let's do more studies in this area. Now, I mentioned that anemia is a major problem or a common problem in infectious diseases, so TB, HIV, malaria, and these are just examples. And why is it a problem? And it's really because of this phenomena of anemia of inflammation. And basically what's happening here is that the infection, the pathogens, elicit cytokines and pro-inflammatory cytokines that actually act on the liver and basically regulates hepcidin. And what happens is that the erythropoietin, which is very important for erythropoiesis, for red blood cells to be developed, that's produced in the kidney, has a blunted effect. It's reduced. It has a blunted effect also on the bone marrow where the erythropoiesis actually happens, and what you end up is with this very, very few red blood cells being produced, very microcytic, very small, and this is what we call anemia of inflammation. This process occurs for it may last up to about 2 weeks, sometimes a little bit longer. Now, this has really important implications in the diagnosis of iron deficiency, and I'll show you in the next slide, but it also has implications on treatment. So with diagnosis, I will use an example, some of the work that I did years ago, where we actually recruited kids who had severe anemia, and we did bone marrow aspirations. And the bone marrow is what you can see on the left, properly stained. This is the gold standard way of diagnosing iron deficiency. And I compared it with biomarkers, iron biomarkers in the blood. And basically what happened, or what we can see, is that these biomarkers are neither here nor there most of the times. As you can see, ferritin has really high specificity, but really poor sensitivity. Look at STFR, another iron biomarker. It has great sensitivity, poor specificity. So what is really happening? What's happening is that these iron biomarkers are also affected by this acute phase response that comes about with inflammation, and it really makes diagnosis of iron deficiency very, very hard. So let's move on to try and look at the effect or the association between iron and infection. So I think the first thing to say clearly is that iron deficiency will have an effect on infection risk, but it does not equal, or it's not just the opposite of the effect of iron supplementation on infection risk. So I will start first by talking a little bit about the first of iron deficiency and the risk of infection. Now, I think the most seminal studies were done ages ago. The one that's really quoted is by Murray and Murray. I believe they're brothers or father and brothers, done looking at refugees that were coming in from Somalia. And they noticed one thing, that those that were iron deficient tended not to have infection, while the iron replete ones had a lot of infections. Now, more recently, going back to the work that my group did a few years ago, again looking at severe anemia, we actually noticed that there are many risk factors for severe anemia, like Epstein-Barr virus, as you can see. There are all these risk factors on this side that actually is associated with severe anemia. While there was only one factor that was protective. In other words, that was associated with less with severe anemia, and that was iron deficiency. So this is more recent proof of this association of iron deficient states being associated with decreased risk. Now, I wanted to go and talk about specific conditions. So I'll start firstly by looking at the association malaria has with iron. And I will talk about iron supplementation in malaria at-risk populations. So these are people in the community who are just generally at risk where malaria is endemic. A huge seminal study, again, carried out in Pemba in Tanzania years ago, set the bar. And basically this study was where the investigators gave iron supplementation to children in the community. And they basically found that the group that received iron had an increased risk of of hospital admissions and also deaths. And a lot of them were malaria related. And as you know, Tanzania is a high malaria transmission area. A similar study, sister study, was done in Nepal where there's very low malaria transmission, and this effect was not seen. And this study really set the scene in terms of WHO giving guidance in terms of how to give iron supplementation in areas where malaria is endemic. And basically said, give it but give it together with effective malaria prevention, diagnosis, and treatment. It's something that we can talk about later on and say, what exactly does that mean? So we move away from malaria at-risk population and come and look at what about iron supplementation in people who have malaria. And these are kids mostly in our setting. I carried out a study a couple of years ago looking at basically asking the question, do kids who have malaria and anemia, do they require iron supplementation? And if they do they need that iron to start immediately? Remember, there's that acute phase reaction that's happening over the two weeks or so, depending on the infection. Do they need that immediately, or should we actually delay the iron supplementation to a time when they're able to absorb more iron? And if we do give iron, is it safe? So I carried out a study where we basically randomized kids who had malaria and anemia. One group, they received iron immediately, 3 days after they had completed their anti-malarial, and another group 2 weeks later, and there was another group that I did not give iron at all. And what did we find? Firstly, whether we started immediately or we delayed the iron supplementation by 2 weeks, there was no difference in terms of the impact on hemoglobin recovery. When we looked at the risk of actually the kids having another attack of malaria or other comorbidities, there was no significant difference in the groups. OPD visits, hospital admissions, or even clinical malaria. We did an iron absorption study looking at stable isotopes. Again, the uptake of iron was similar in the two groups at the beginning and even at the end of the study. Another study done by colleagues Chandi et al. In Uganda, now looking at still malaria, but now looking at severe malaria, so kids with cerebral malaria, kids with severe anemia and malaria. Again, delayed versus giving iron immediately. The results were the same. There was no significant difference between these two groups. And even the risk for infection, another malaria attack, was not different in the two groups. So really, what can we say from these two studies? And I should say that these are the only two studies that have been done in this area. So there's a paucity of data. But for prevention, if we're giving malaria, iron supplementation to the community, we have to be careful. We have to give it with good malaria prevention strategies. If we are treating, using iron supplementation to treat anemia in malaria, we probably don't need it. I'll just move on very quickly to TB. There's no studies that have been done, trials looking at iron supplementation and its effect or association or link with TB. The only study that has been done is by Zimmerman et al., Michael in Switzerland, and they were working with a study where they did a really beautiful workup of the iron status, hemoglobin status, inflammation status of these TB patients before treatment and after studies looking at iron absorption from the gut. And basically, to summarize, what they found was that it was the same in TB, that the central problem or the central mechanism for the anemia was anemia of inflammation. So they showed that there was a high hepcidin in the beginning, which was leading to this anemia of inflammation, and that after that initial treatment with TB drugs, this resolved. And even the uptake, iron uptake, actually improved with treatment. So what really can we summarize from this is that probably we should withhold iron supplementation in patients that have TB and anemia until the intense treatment phase has been completed. With all these diseases, there are actually no treatment guidelines, or there are very little said in terms of the treatment guidelines how you should treat anemia in these diseases. So that's another big area that needs to be tackled. Lastly, I'll just talk about iron and HIV infection. And again, there's basically no trial apart from this one that I did where I supplemented children who were HIV infected and anemic, mild to moderate anemia. And this was a time before they weren't all put on ARVs. It depended on your CD4 count at the time. And basically randomized them to get iron and another group placebo. And what we found was a couple of things. So this is a forest plot that shows you, on the left, if the estimates fall on the left, this is in favor of iron supplementation, while on the right, it is basically not in favor. And basically, iron supplementation in this group, it reduced hospital admissions by 30%. HIV progression measured by CD4 count, it also reduced it by 34%. It was protective. HIV progression, there's a typo, but respiratory infections were also reduced by 41%. The only thing that there was an increased risk was malaria. So what can we really summarize from this study? Probably iron in this patient group is beneficial. But again, we have to be very careful with malaria, and we have to give iron supplementation with adequate malaria prevention and treatment strategies. And one of them is providing a bed net, which has proven to be quite beneficial and good. And so, coming to, this is my last slide, I think, some reflections on nutrition and infection, and especially iron deficiency and iron supplementation. One, I think it's quite clear that we should look for the etiology of anemia before jumping and giving iron supplementation. I think it's quite clear even from some of the work that Andrew just presented, where they gave iron and it did not have as big an impact on the hemoglobin. It improved the iron status, yes, but it tells us that there's a lot of other things happening, other causes of anemia. I think other areas that need for us to really look at a little bit more closely in terms of research gaps, I think is looking at the link between iron and iron supplementation of other forms and the risk of infection. I think also we need to look closely about looking at the effect of iron supplementation in pregnancy and also in severe acute malnutrition. for those who really want to, work in these areas. And lastly, I think it's the link between iron supplementation and cognitive function, which Andrew, again, quite rightly articulated. So we know that iron deficiency is associated with cognitive dysfunction, let's say, but we don't know whether if we do give iron supplementation, we actually reverse that. So I'm presently carrying out a large study in Malawi to try to answer that question. Lastly, I would just like to thank various investigators and also funders for this work that spans over a period of about 10 years. Thank you.
Thank you very much, Professor Firi. Let me just add that I don't know what's happening in the Gambia, but we have seen from the last DHS, this is 2013 DHS, to the 2018-19 DHS, as I won't say significant, because before the statisticians strangle me. We have seen a massive reduction of anemia amongst women of childbearing age and children, to the tune of about 30% for both groups. We don't know exactly what is happening, but it's food for thought for us. Malaria may be the reduction in malaria. It may be an improvement in something else, but we don't know. But there is something good, or a number of things good, that we are doing. We hope that the next DHS will be able to give better results. Thank you. And on that note, I have the pleasure to invite Dr. Chris Murray from the MRC Gambia, London School of Hygiene, the planetary health. We all know that climate change and nutrition is a new thing coming up, but it's something that we should be worried about. And I have the pleasure to ask you to educate us a little bit. Thank you, Chris.
Okay, yeah, thank you. Thank you so much for the introduction. Assalamu alaikum. So, yeah, I have the kind of unfortunate job of often bringing some of the kind of worst news to meetings like this. It's been so amazing to hear about, just the fantastic achievements and the successes in health research attributable to the MRC and the incredible researchers that have been here for such a long time. I lead our program in planetary health at the MRC unit, which I've affectionately started to call the miserable prophets of Doom Lab, and as we constantly come in and essentially just ruin the party often. So I just wanted to sort of try to use this time to kind of reverse that perception a little bit and tell you about some of the stuff that we're doing that is really sort of positive and proactive in trying to sort of combat some of the bad news stories that you've been hearing. I am, however, going to start by ruining the party and talk about a few different areas which I think are just inseparable from this, the whole the issues that we're talking about in the space of climate change, nutrition, infection, and the types of work that we're going to be doing at the MRC unit. So this is a couple of quotes that I pulled from a paper published in 1953 by a guy called John Clark, an ecologist who was part of an Oxford University expedition to the Gambia, conducting surveys on the hippopotamus in the Gambia River. And at that time, the coat of arms of the Gambia was this beautiful trumpeting elephant in front of a palm tree, which somewhat ironically was last seen in the Gambia, or at least last shot in the Gambia in 1913, some years ahead. And by the time that the unit was established, there were still lions in the Gambia, although Clark comments that they're regarded as quite scarce. Nevertheless, this is just an indication of how much has changed in terms of the wildlife composition of the country. And so it's also somewhat ironic that the second coat of arms adopted in 1964, coinciding with independence, swapped out the elephants for these two lions, holding an axe and a hoe, and reportedly representing this colonial history and the future and importance of agriculture to this country. And now we've, if we fast forward the clock, we've not only lost lions and elephants in this country, but around about 13 of the largest mammals in this country are no longer found here. And it's really just a tip of the iceberg if we're thinking about biodiversity loss. Turning to climate, we got a little bit of an introduction from Andy Haynes about this, and again, I'm very sorry to say, but the story is as told. There's really no way around it. If you were one of the founding scientists at the MRC, you inhabited a space that was very different to the place that we inhabit now here. The climate was on average around about 1.5 degrees cooler in 1950 than it is now, and you can see from those charts that increase in average temperature as well as that change in seasonality. Of course, the rainfall's also changing, and what that means is that the wet seasons that the initial scientists at the MRC experienced were also considerably wetter than they are now, around about 30 or 40% wetter. And you can see the concentration of drier wet season in the more recent decade. So this landscape is really fundamentally changing from a climate perspective over this relatively short period of time. If we look at the projections, which are those African maps for the future, the top one's for the end of the century under a strong emissions or a no emissions cut scenario, what we call a business as usual scenario. We're looking at warming of something in the order of 3 to 5 degrees. Now, I don't think we'll get quite that bad, but there is nowhere in Africa that's going to be any cooler than it is now, and very likely it'll be in the order of, another 1 to 3 degrees from where we are now. So blowing well past the Paris Agreement, which in this part of the world is where we're already at right now. Precipitation, much more difficult to summarize, but basically there are areas that are going to be wetter, there are areas that are going to be drier, and one of the areas that is going to be drier is this area right here, and that's related to the drift of the intertropical, basically the wet season, the weather pattern that comes annually that brings the wet season is slowly drifting south. So we're on the top margin of where this system delivers rainfall in the wet season, and it's gradually drifting south as a result of climate change. Turning now to what things look like on the ground here. This is a picture, a satellite image from 1973, so this predates me by quite a few years. It's a bit fuzzy and it's artificially coloured, so it looks really green, but nevertheless it's pretty clear that there's still extensive areas of vegetation cover, even in this area of the Greater Banjul area. If we flick through just a few time points, this is 1985, this one is 2016, and the most recent one I have is from 2021, and you can see really there's basically nothing left except for that massive area of mangroves there, which we still haven't, there's still been relatively little development in there. If you zoom in a little bit, one of the very few patches of good quality forest that's left in the Gambia is this patch right here. The astute amongst you, or perhaps the pre-prepared, or perhaps those that have gone for a bit of a wander at lunchtime will recognise that yellow star as the place that you're sitting right now. This is the auditorium of this conference centre, which was built in 2017, directly on this tiny fragment of forest that's been clinging on under the protection of a forest reserve since 1952. That represents about a 20% loss of this area in the space of about 2 months. And we've just heard news in the last couple of weeks of another development that's being proposed in this area here, another 10 hectares. And this is really just to illustrate that this is a really creeping process. Everyone's implicated in some way, shape or form in this process. And it's representative of what's happening at a national scale. So this, the figures on the left there are showing land use change maps from 1985 through to more or less the present day, and the stats are similar. It's about 20% deforestation, loss of natural cover in this region. If you go back to 1950, the founding of the MRC, it's about 50% of the entire country has had a change in land use representing this vast deforestation. The country looks completely different than it did back then. Of course, these are the types of issues that are really high on the agenda for the planetary health community. We were perhaps cosmically censored in Andy Haynes's eloquent talk about this, but he literally is one of the people that wrote the book for this field. And really the kind of key sort of messaging in this field is that we really need to start thinking much more upstream, much more big system, much more interconnected processes that underpin both ill health and health protection for the future. And this is what our group is here to try to do. Umberto showed this figure yesterday in the description of the MRC structures, and I promise this is the last Venn diagram that you'll see in my talk today, somehow representing planetary health into, that touches upon all of the existing themes here, and I'm going to talk a little bit about some of our work in that space. And I'd also use this slide to draw the link to the Centre on Climate Change and Planetary Health, one of the 13 centres at the London School of Hygiene and Tropical Medicine, of which I've recently become a co-director. And the agendas that we have at the centre and in the MRC unit in the Gambia are similar, I would say, not exactly identical, but very similar. So we draw great strength and we draw great inspiration from people like Andy Haynes, people like Alan Dangor, who was the previous director of the centre, that have really raised the profile of planetary health issues in the last few years. Okay, I lied about the Venn diagram. I'm very sorry about that. This is roughly how I envisage the current activities at the MRC unit in planetary health. It consists of sort of 3 priority areas at this stage that more or less reflects the activities of the people within the group. And I'll just sort of talk through a couple of those different areas. So I'm going to start at first with climate change. This really needs no introduction. We had Andy Haynes today, we had Anna Bonnell yesterday, pushing us off a cliff of despair with her studies, pregnant farmers in rural areas in the Gambia. Really unbelievable statistics if you consider that basically nobody went out to check, and it's really dramatic the impact that Anna's work has been having in this space for our group. So we're studying the direct impacts such as heat, but also some of the more complex impacts of climate change, for example through the effects of rainfall on diarrheal diseases and nutrition, and we have quite an interest in an area that Andy really canvassed very well on the health co-benefits of some of the solutions to climate change, what we call the health co-benefits of climate change mitigation. Because this stuff is happening all the time, it's going to happen anyway. The world is very well aware we need to do something about climate change, and health should really not be left behind in this discussion. And so this is an area where we see great potential for health, not only to feed into that discussion and make it stronger, but also to lock in health benefits of these activities for the future. Finally, in this space we have a very interesting development that we'll host a African regional hub of a really ginormous global project called the Lancet Countdown on Climate Change and Health. It's a project that I've been a part of now for 5 or 6 years that compiles information around progress towards the Paris Agreement from a health perspective using presently I think 43 quantitative indicators that track year on year how we're doing with that ambition. So here just shows one indicator. It's an impact indicator showing the frequency of drought. We have 43 like this, and it gives us this really overall picture of what's happening in this climate change and health space. We published the report, the 2022 report, about 2 weeks ago in the lead up to the COP. And alongside that, there was a European, a South American, a China, and an Australian specific Lancet Countdown report. And next year, our ambition is to have an Africa-specific Lancet Countdown report to coincide as well. The nutrition and sustainable food systems theme, we're considering issues like dietary diversity, the ecological integrity of food systems, and of course land use change. What I didn't mention earlier when discussing land use change is that food is by far and away the most important issue when it comes to what the planet looks like. Farmland area or agricultural areas occupy roughly 50% of the habitable terrestrial land surface, and in some countries it's even higher than that. It also uses 70% of the world's fresh water and is the #1 cause of biodiversity loss. So it's absolutely fundamental in this discussion around preservation of ecosystems, but also protection of health through food security and nutrition. We have some preliminary work looking at, for example, the diversity of landscapes and the diversity of food-producing landscapes and how that translates to the diversity of diets, and through that mechanism, health of people as well. And we're very interested in the restoration agenda because, of course, a lot of this has to be reversed. There's a lot of landscape restoration occurring that will occur with or without us. For example, lost ecosystems across the entire width of Africa as part of the Great Green Wall. Some of that is around restoring landscapes through agricultural development, so selective plantings of food-producing species alongside other species we find out. Finally, the third circle is the infectious disease dynamics, or the One Health kind of circle. This is kind of the eco-epidemiology of infection, for want of a better term. My preferred area of this very complex interaction of different project ideas, where we're thinking very much about zoonotic diseases, emerging infection, the stats that I was mentioning on biodiversity loss and the loss of habitats, the transformation of ecological systems. It's very clear that the root of many of the infections dynamics of vectors. It completely reorganizes the distribution of zoonotic hosts. And through this work, we're trying to, with the climate, to try to anticipate. Here, Aliou gave a great introduction yesterday of his work looking at the influence of agricultural development on human-animal contact, which is probably the most cited mechanism for zoonotic spillover, particularly related to land use change, but the least quantified, the least researched, the least understood. We really have no idea what conversion of landscapes does with respect to changing the contact rate between zoonotic hosts and people, or the bridge vectors in between. Another example is a biological model there that I've shown of the mosquito Aedes aegypti, which carries a number of devastating arboviruses. We use biological model connected to a climate model to try to anticipate whether the future climate will be more or less suitable and where. And we can see from this work, the map, global map in the bottom left, that for the most part the world is getting more suitable for this vector. Spatial variation in that, of course, but we can use models like this to really try to sort of pinpoint hotspots of perhaps emerging or re-emerging infections. And the top, or the central panel there shows the same model adapted to the sister species Aedes albopictus, also transmitting a number of horrible diseases, but at a very, very micro, in this case, deforestation for palm oil in Malaysia. Both of those outcomes, the macroclimatic change and the microclimatic change, ends up benefiting the vectors to around about the same degree. The difference is the land use change does it. Linkage areas as well, and I just want to talk briefly about some of those biggest. I previously thought it was the single biggest or the second biggest employer in the Gambia. It's a household name and universally seen as, viewed very positively. Everybody is Gambia. We should be leading by example. That was led by a bunch of students in our group, that won the Queen's Platinum Jubilee Award for innovation in driving institutions towards a net zero carbon footprint. It involves recycling, grinding it down into otherwise have to purchase, and it's a scale up of an idea that already exists here. We have already good proof of principle that we can do it. We just want to make it circular and more sensible from a sustainability perspective. Connected to that is the processing of organic waste, and instead of incinerating it, we want to use it in a biodigester to produce biogas, which we can use in the Banta Bar, which I'm sure many of you have visited while you've been on campus. And a third example, the most recent, is we've just received funding with Ed Clark as the PI of this project through a Medical Research Council Environmental Sustainability Award to trial the use of drones to divert the use of our 4x4 fleet for simple tasks like carrying. And the proof, the business case of this, of course, is if we can divert a considerable amount of these journeys and replace the diesel-powered transportation by solar-powered drone journeys, then we're really making a fantastic contribution that not only reduces the direct effects of the pollution from those vehicles, but also contributes to eliminating our own source of dangerous pollutants that we know is affecting the health of the population here. Components, so we held a planetary health conference in February, January 2020. This has really sort of been a first step. Change Solutions Festival in May last year that brought together school students and NGOs from all across the country to discuss and learn about climate change and environmental, which is one of the solutions that was identified at the Climate Change Solutions Festival by communities as being an interesting and potentially applicable solution to the collection and the overcollection air pollutants involved in using that firewood for household cooking. I'm going to skip this slide because Andy Haynes already showed it, although it was slightly disrupted. And I'll just go to my final slide, which is, again, something we've heard time and time again in this meeting, is that there is this black hole of information that continues to exist in Africa. The same is true for planetary health research. There's a couple of reviews that have shown that planetary health research is still not penetrating as deeply as we need into Africa and other areas. The same is true for One Health, the related recent purple gaps for West Africa.
The festival you held was very educative. We were there and we were very impressed. Actually, we are into salt iodized season for the consumption of iodized salt, and we saw a school that was doing things for the population. So now have the honor to invite my own very brother, Dr. Job Modu. Modu is doing a great favor for the Gambia by being a young scientist getting into, there's a lot we also don't know about NCDs in this country. And you hardly go to any compound in this country today and you don't hear of somebody complaining about having one or two, and they start explaining. You wonder where they're getting that. MRC, the Gambia, is getting into this and allow us to know what we don't know and can then translate that into policies and programs. So Modu, thank you. Let's hear from you.
Thank you very much, my brother Ahmad, for your words of introduction on non-communicable diseases, which is now part of the unit's work. In the next 20 minutes, what I intend to do is just to give a brief background on the burden and drivers of NCDs, what about our rationale, and before giving briefly on the overview, on overview of some research projects that we have undertaken before concluding with what we have found so far. There is a high burden of, and I just want to give two examples. One is on hypertension, that we see almost a linear increase in the prevalence of hypertension in sub-Saharan Africa. Whereas in 1990, we had about 50 million people affected across the sub-region. And similarly, we can say something like that for diabetes and impaired glucose tolerance. The Gambia actually is no exception, and yesterday the director of the 20% of the country urbanized in the 1970s when we became a republic, to now about 60% of the population living in urban settings. So this is really a stark reminder of what might happen. And then we also see a lot of changes, patterns, the proportion of older people as a result of increasing life expectancy, as well as, the changing disease patterns. Here we come, and this we have seen compared to 2009 and 2019. So majorly, we now see ischemic heart disease, and this is, as I said, for the whole population, children included. That ischemic heart disease is now the second largest cause of death in the country. Stroke has increased in rank from 8th to 5th position. And just not that, but also the percentage increase that you see for each of these conditions. Whereas other diseases almost remain where they were, 0.6% change for respiratory tract infection, and stroke about 34.8%. I didn't highlight for cancers, but you can also see something similar. So as a result, at the unit, we have been enhancing research on nutrition-related chronic conditions. And this we try to do as much as possible with several government departments. And the National Nutrition Agency being one of them. The director is my able chair here. And also a two-way relationship, especially the non-communicable disease program. They've been increasingly inviting us to participate in their activities, especially when it comes to research, as well as technical advice. And we also involve them as to how our research can make more impact before even we start the studies. And also with the Gambia Bureau of Statistics. Briefly on the drivers, as we can see, this can be taken to almost all settings. Social lifestyle factors. What do we do in our context? And we ask ourselves always just these two questions. That are there specific characteristics of these conditions that we are trying to address in the Gambia, in the sub-region, and across Africa? And the second question is, can we better understand this in order to develop Africa-specific interventions and therapeutics? And the rationale for this is most of the treatment guidelines we use, and I give here an example of hypertension, is mainly relying on data collected elsewhere, and mainly from black populations in the US or the Caribbean or elsewhere. And even for countries in Africa where they've developed their own guidelines for treatment, they extrapolate data from these settings. And I quote, that hypertension epidemic as patients. In contrast, they extrapolate from US data. North American and European black population, vascular risk and response to an antihypertensive drug treatment. And I think the same thing applies in our context. So we try to do research answering those two questions that I have previously mentioned. Hence, we do research in all these areas from understanding the mechanisms of the diseases, and so far, obesity, diabetes, hypertension, sarcopenia, and multimorbidity. But it's more than this, but these are our main focus areas for now. Talking of sarcopenia, is led by our dear friend, Professor Kate Watt, who is based in Southampton. Just a brief overview of some of the projects that we have undertaken so far. The medium study, in which we try to understand diet, microbiome, adipose tissues interactions through the mechanisms of metabolic endotoxemia that I'll come to in a bit. The need for more genotyping. Here we targeted individuals with variants of the MTHFR C6 high-dose riboflavin. So know that individuals deficient in riboflavin also tend to have bigger problem in controlling their blood pressures. So we did that for the rural Key and West population, where we have evidence of this, riboflavin deficiency. And we hope the results will inform us something about why it's difficult to control hypertension. In a study in which is a clinical trial almost completing, in which we try to understand whether simple lifestyle methods like physical activity or taking of yogurt can help reduce gestational diabetes in pregnant women. Another study that I just want to highlight is the hypertension, for example, the study in which we are following up participants doing really in-depth measurements, see if we can even diagnose, which was previously definitely for cardiac electrical activity. So briefly, just to highlight the medium study, as I said, here we try to understand how do diabetes occur, especially in our context. And the hypothesis was that gut microbial products translocate across the gut wall. And this can be, and when this translocates, the LPS processes such that inflammatory cytokines that are produced would impair the binding of insulin to its receptor, or that the insulin receptor itself will not be expressed properly. And this leads, we studied that in urban contexts, recruiting 3 groups of women. And in each of these groups, we measured, for example, gut permeability, gut biopsies, and so on and so forth. So far, we've recently published this, and what we found was that, that there was no difference in mannitol assays. And it was similar across the groups, lean, obese, and obese diabetic women. And with this, also what we found was that just a confirmation from an earlier study by a group that LPS, non-diabetic populations, in core antibodies. And here we concluded by, with that obese individuals have a constitutive deficit in binding and being overwhelmed, and LPS levels rise, driving insulin resistance. And this is opening another possibility for research in the near future. Just to show you, or just to reiterate what the chair of the session said, the prevalence of diabetes. So we undertook, embedded an NCD component in a national survey that was led by the International Center for Eye Health at the London School in collaboration with the Bureau of Statistics. And this was in populations older than 35 years of age. And what we found was really something we've never seen in the country before, that the overall prevalence of hypertension in men was about 44.7%, and in women, nearly half, and that's about 49.3%. And we saw this with subclinical organ is a possibility of bigger complications to happen in the near future. if left unchecked. And similarly for diabetes, almost higher than what we see as an average for the region. 5.6% of men and 7% of women having diabetes. Again, in terms of body mass, using body mass index, we saw really higher prevalence of obesity and overweight in women across all men, 20.3% and 4.1% in women and men respectively. The difference was actually higher in urban compared to rural, but nonetheless similar pattern. I just want to show you when we looked at the cascade of care in terms of hypertension. So if we see men with hypertension, which is the figure on the upper left of the screen, so out of all men who were hypertensive, only about 43.6% were previously diagnosed, according to the survey. And of those who were diagnosed, 29.4% were treated, and out of those who were treated, only 6% had the target BP control of 140/90. So something is not working, and this is across the cascade. And we can also see similar for women, slightly better in terms of control, but it's almost the same that there and those controlled. So we need to understand a lot more in terms of the cascade. So with this, we come to terms of blood pressure. They knew they had hypertension, but they never took the treatment for whatever reason. Those who were hypertensive and were receiving treatment, and those who were hypertensive to understand the pattern, others compared to those who were untreated and unaware. And this in men was more severe compared to women. And we've been trying to understand what is happening, and perhaps there can be more room for discussion around that. But it could be still that there is an adherence problem, or the health system, whether the way we are treating needs a rethink. And this is just for systolic BP, and it was just a similar pattern for diastolic BP as well. So we think that there is a need to enhance screening to improve diagnosis rate. For the untreated, at least there should be greater allocation of treatment. And for the treated individuals, so far, based on what we have, is just to reinforce the treatment emphasis, playing emphasis on lifestyle intervention, and revisiting current pharmacological treatment guidelines. So this is really something worrying, and as a response, we just got funding from the NIHR to start this improving hypertension control in rural sub-Saharan Africa, and this project has just, we'll just kickstart after ethics approval, and it's taking place both in our rural Kiang West site as well as in Kilifi in Kenya. It's mainly trying to understand the health system, trying to map a typical patient, be speaking with policymakers, healthcare providers, so on and so forth, and just trying to understand the context we are dealing with. And the second work package, which I'll be leading, is trying to improve the diagnosis. And this project, I forgot just to mention that it is embedded in the normal health structure that we have in the country, using community health nurses and village health workers who will implement the project. Measurement types with of these three will provide the best means for diagnosing hypertension. But we'll also want to measure the burden of target organ damage in the population, as well as using point-of-care testing to see how well it will diagnose target organ damage for participants. So it's all about trying to improve risk stratification. And using the lessons we learn, we will be learning from work package 1 and 2, then we will set up. We also have a strong training, capacity strengthening that will be led by that is led by Professor Asenjay. And it's all also, we've been doing this with the regional health team especially. Team leader for his encouragement, our collaborators and funders. Thank you.
Chris started by saying he was having to say that those who are hypertensive and under treatment are not even doing better than those who are not treated. That's scary news for some of us. But yes, this is just to demonstrate. Andrew has said in his presentation that nutrition status is improving in Africa. And I note that nutrition able to have stunting from 17%. Sometimes some reports will even report 14%. So that's about half stunting. So we're doing well, but we know that we are also sitting on a time bomb when it comes to diet-related non-communicable diseases, especially with hypertension in the Gambia. And on that note, I wish to open the floor for a few questions. We're running late, so it will be just a few questions before we thank our panelists. I have to take a mic so people can hear your question.
I want to thank the presenters for the brilliant presentations. And I would like to have a question for Dr. Mudu. I want to find out from you whether the reference values you used for hypertension experiments we're conducting, of those who had hypertension, who were aware that they were hypertensive, and those who were not aware. Was there any effort to desensitize or debrief the patients after their initial, after raising their awareness or after their initial test? Because it seems as if at times when you indicate that a patient or he or she is sensitized, and it may even be, get the person to become more worried and develop even more problems. Is there have you factored this one into your research, where there is supposed to be desensitization? Thank you.
Just the last bit there is what I missed, whether we have, after desensitization, sorry about that. Okay.
Do you make an effort to help the patient, to explain or to help him come out of the situation? Because normally, in some cases, when you tell somebody that he's hypertensive, it may trigger other reactions that may even worsen the situation. How do you handle this in the research team?
Yeah. Okay. Thank you. Thanks for your question. In terms of the whether we have any local reference values for diagnosing hypertension, this we developed in pediatric populations, those who are up to under 17 years of age. So we compared this with the US fourth report, and using that, we would have been estimating the prevalence in our settings. So that was just to measure blood pressure, age, and some anthropometric parameters. But for adults, we used the cutoff of 140/90 still for the diagnosis. So for this survey, it was evidence of receiving medication and or having values higher than the threshold that I've mentioned. So in terms of participants so this is more for a clinical setting, your question, I believe, whether after diagnosis, do we make an effort and how do we handle the situation? There, I believe, so for us, it was more of measuring BP and referring the participants as and when needed based on the values we record. But we also offer counseling, lifestyle intervention, and so on and so forth. So those are just some of the things we do. But I'm pretty much aware that care may not be optimal in that area because of the numbers and the healthcare providers that we will have available in the facilities, such that there is very little time between patient and whoever, doctor or nurse contact. So perhaps that is not optimal, if I got your question right. Thanks.
Thank you all for the lovely presentations. My question is to Chris. How sustainable is the solar cooking for day-to-day living? Like, how sustainable is that?
We are struggling to get you, so maybe either they increase the volume or you increase your volume. Thank you.
I'll try and scream. How sustainable is solar cooking? Can it be used for day-to-day, lifestyle? How sustainable is that?
Yeah, at the moment, I don't think we know. That's part of the reason why we want to hold this solar cooker competition, in order to really kind of nut out the practicalities of something like this. It's very clear that for the air pollution related to cooking with biomass, particularly wood and coal, we know that's a terrible idea for health. We also know that firewood collection for conversion to charcoal or for use itself is also terrible for the environment when it becomes to such a degree that it's beyond the capacity of that environment to replenish itself. So there's a win-win there potentially, but there are costs as well. The costs include people being unfamiliar with that. The time, it's definitely not as instant source of heat. And I guess we have to admit, Anna, that in our trial of the first prototype of the solar cooker that we received on the weekend, I don't think we barely had any cooking occurring in our pot whatsoever. So there's a huge amount of learning that needs to be done to work it out. The reason that we're interested in this particular solution is because it was nominated by the communities that were involved in our Climate Change Solutions Festival. They identified that as being something that they wanted to try out, something that they thought could have good potential, and we're just very happy to try to facilitate that. We probably need some engineers involved. We definitely need a lot of trial and error, but let's hope we get somewhere.
Thank you, Chris. There is one from this side. Okay, no, let's get the one with the T-shirt, black and white T-shirt. On the extreme end, Mr. Mike.
Presenters, I think it was really a great presentation. My question goes to Modi Jobe and those that have hypertension. The treatment group are doing less better than those that are not on drug. So, can you hear me?
Oh, come again. We're struggling to hear you from this end.
Yeah, what I'm trying to say, it's really ironical to see that the treatment group is doing less better than those on treatment in terms of those that have hypertension. So I don't know if you try to explore further, what could this two group, what is the treatment group doing that is those that are not on treatment, what are they doing that this treatment group are not doing? Maybe you want to explore further, are they adhering to the non-pharmaceutical guideline or treatment better than those on treatment? Or those that are on treatment think that the pharmaceutical treatment suffice the non-pharmaceutical treatment, maybe better than those on treatment. So I don't know if you understand my question. So my second question goes, still to the module. There is this dogma with regards to those that have hypertension in terms of the non-pharmaceutical guideline or treatment. Maybe the clinicians will tell someone to consume less salt, or you don't consume salt, and alternatively people will tell you to consume the food sweeteners, that is the jumbo and co. So I don't know if there are evidence to support this. Maybe you can try to shed more light on that. Then the third question goes to, sorry, apology, I can't remember, in the first presenter. Yes.
Professor Firi. Yeah. Good.
Yeah, my first question goes to the first presenter. They try to establish a relationship between anemia and infection. So, though you try to establish that iron supplementation might not necessarily be needed in patients with severe anemia. So, in African setting, diagnosing iron or trying to establish diagnosing iron deficiency is really difficult, especially when you go to the rural communities and stuff like that. And when patients present with severe anemia, acutely they get blood transfusion. So meaning most of those bloods are coming from the blood transfusion. Most of those ions are coming from the blood transfusion they receive. So how do we do, or what could be an intervention point in terms of the blood transfusion that these patients are receiving to see that the iron contents are reduced? Is it possible we have a point of care diagnostic test for iron, since we are in the period of technological advancement, if this could help? Thank you.
Thank you very much for your questions. So for the treated patients, we can only speculate as of now. We don't know exactly, but there are possible explanations to this. One is perhaps they had more severe hypertension, which is why they are detected and under treatment. Secondly, it's that they may have other risk factors, which is making their blood pressures harder to control. Perhaps obesity, smoking, and other factors may be in them. So a lot of factors that we are exploring at the moment. For non-pharmacological treatment, I agree. So there is, a lot of evidence out there to say that other lifestyle factors, like reducing body weight, stopping smoking, etc., can't help reduce the blood pressures up to a certain level compared to even a pharmacological treatment. So the two should go hand in hand. So what this calls for is there is a lot to be done in terms of the care of the patients using both means.
Thank you.
All right. So thanks for that question. I think you've asked the billion-dollar question for us who work in iron deficiency. Point of care test. That would be the most useful tool that we could use to actually even tackle anemia. I actually was in discussion with a manufacturing company, and they're talking about how do we do, an RDT for malaria, and they sort of asked me what other tool would really change the landscape of, anemia. And a point-of-care test for iron deficiency would do that. It's very hard. Most of the biomarkers that we use take time to measure. They are expensive. They require equipment that would not necessarily be there in most secondary health facilities, definitely not in primary health facilities. So it is really difficult to measure iron deficiency in that setting. And if you do even have the capacity to get those biomarkers in areas where there's infection, as I showed, it's really hard to interpret your finding. And I think that's why in the end people tend to just move on to give iron supplementation. And basically what we're trying to say is that, it probably would not be helpful, and it might actually be doing some harm.
Thank you very much, Professor Fury. Actually, iron deficiency biomarker, during my PhD, we were trying with hepcidin. The hepcidin guru is here. Maybe you can talk to him and see where we ended with use of hepcidin as a biomarker for iron deficiency. I can see hands flying. Hassan, can we take one or more? One more. Hassan, give me two. Okay, one here. Quick one.
Yes, this is a quick question to Badu. Because as you mentioned, we have noticed clearly that there is an increase of non-communicable disease right now. And in one of your slides, you have shown that there is a genetic factor related. I'm just curious. I want to know if there is a specific population in a specific manner you have found that the genetic is related to hypertension or diabetes.
Before you respond, let me just take the other question. Yes.
This goes to Chris. That was a very good comprehensive talk. We all observed the little power crisis that we were just having here. And from your presentation, Gambia is actually expanding and developing, which is leading to more urbanization. So the kind of data that you are generating, I want to know how you are liaising with the government and policy so that more of the rural areas are going to be urbanized. So how is this being integrated so that there can be other alternative energy resources being made available? Gambia is close to the sea, so it's likely you might want to look at not just the solar, but tidal waves as well. So it's just a little question that I want to ask. Thank you.
Quick response. So as my boss mentioned, APOL1, Apple, is one example. And the other is what I mentioned in my talk, the MTHFR C677T gene. So for that, for sure, most likely they may not respond to medication, but what they will respond to would be riboflavin supplementation. So that's something, just a quick response.
On the issue of sustainable development in the Gambia, really, I think is the kind of umbrella of your question there. One thing that really struck me in Andrew's talk was this, the sort of very generalizable response from the nutrition perspective on socioeconomic development as itself a multifaceted driver of improved health. This, of course, has been the subject of a huge number of development projects, not just in the Gambia, but all over the world. In some cases, that is shown to be really good and really good for people and really good for populations, even their health, their finances, livelihoods, all that kind of stuff. There's an interesting study in the Gambia that was, I think, from the World Bank, where they did a really large randomized study of an intervention supplementing populations with cash, basically. And it had very good social benefits, but it came at the expense of the surrounding environments. In the villages that received the treatment, you had a degradation of forest resources, you had an extraction that was higher of ecosystem goods and services as measured from satellite compared to control villages that didn't get the treatment. And the key here is that there is often this trade-off between, the environmental reserves that we use to supplement our lives and the benefits that you derive from that socioeconomic development process. In the Gambia, we have a huge opportunity to leapfrog many of the mistakes of populations of the past by just not going down those routes, just not going down, for example, or leapfrogging directly to new technology. So you mentioned the installation of solar, for example, makes brilliant sense in the Gambia, provided that we have the technical capacity to install and maintain those types of technologies. So I would say the key thing is that we just don't keep replaying the same old tape, that development is a very complex process, but it can't come at the expense of the environment. Let's leapfrog just to the sensible and smart solutions that we know are being tested around the world and should be, as far as possible, pursued here as well. Thanks.
I quite agree with the leapfrogging. There is a lot of mistakes that we in the Gambia can avoid and get our indicators right without making the same mistakes. And some of our institutions are really focused into that, and we are piggybacking, or we are borrowing from the research to make them our policies and programs. Yeah, I wish to thank my eminent panelists. I think you have done justice to your subject areas. Professor Firi, thank you very much. Chris, thank you very much. Modu, thanks a lot. And
thank you very much. I think overall today we had a very great session. I think apart from the small hitch we had with the electricity, and we apologize for that. I do hope that tomorrow we won't encounter that. We've registered our complaint and protest to the administrators here, but they said it's not their fault, but I happened to bump into the company's director who was attending a meeting and when this thing was happening. So, and I told him that this is unacceptable. Anyway, we hope it won't happen tomorrow, but overall it was a very great session. I think we all enjoyed the sessions, very fantastic presentations, quite fruitful discussions, and I'm sure all of you enjoyed it. We're sorry, I think at the later end I saw many people dozing. We should have perhaps brought the coffee earlier than after the program. So, but in any case, there's coffee outside, I think, this way, and we should hurriedly get the coffee and then get to the buses so that we freshen up to come back for the banquet dinner, which should start at 8 o'clock. So we have limited time, about almost only 2 hours, I think, to go and freshen up and join the bus. So the police escort are going to wait and then escort you back here in the evening, because that evening is really a bad traffic time. So the police escort will will stay on the hotel until you people are freshen up and then get you back here by 7:30. Somebody lost a phone. I think Samsung, in case anybody come across it, please just bring it forward. So we really want to thank you all, and I hope all of you enjoyed today's scientific session, and it was a great day for all of you, and hopefully we will have the similar great sessions tomorrow. The final announcement is that there are some people who've got these 2 copies of MRC's report about our structure, about our science, summary of the science, and the summary of the platform. But we decided to give it only to the, international scientists who've traveled to come. But I don't think all of them got it. So what we've decided to do is to copy them into USB. And we have lots of that USB, which we will distribute tomorrow. Even those who have the hard copy and would want to have the soft copy, we will still give you, because I think this is a lot anyway. The USB is a lot. We wanted to copy the presentations in it, but we couldn't get, the presentations, all the presentations, early for our IT to do it. And because they are about 500 USBs, they felt there was no time to do it. So I'm sorry, what they have suggested is that they will put, for those who've given permission to share the presentations, they will put them into a special link, and people will access the link and download them. Is that fine? But rather than, putting them into the USB. Okay? So with that, I thank you I thank you on behalf of the organizing committee and the MRC leadership board. I thank you very much for your patience, and have a nice evening, and let's hope to get a nice banquet dinner. Thank you.
Okay. Oh, what a long session.
Great.
Thank you.
Good session. Yeah.
Thank you. Chris,
Good to actually
we've been
in the flesh.
Yes, in the flesh. Good. Emails have been flurry of emails. I've not seen Chris. I complained about that. I said, Chris, you should come and see us.
I think we actually need to organize a date for that.
Certainly. Yes.
Vamos.